Thank you for giving me an opportunity to talk about some of my work. I think it's been set up pretty well by some of the previous talks. Dr. Walston talking about mitochondria, and it's come up in a number of different areas. And I think as everyone remembers from their, 8th grade biology class, 9th grade biology class, mitochondria are the powerhouse of the cell. And so really thinking about with respect to aging and healthy aging, there's the role of energy has a critical role. And if we think about this, you sort of have this model where you have a certain amount of available energy and you have a certain amount that's required for homeostasis or sort of for everyday living. And then you have some sort of stress in your life. You decrease the amount of available energy. You use more for just maintaining your function. And then normally you'd recover and you'd have the same amount of available energy. You'd use the same amount for homeostasis, so you dial things back. And what we find that in aging, that there is this sort of dysregulation. So you essentially are unable to revert back to your normal state, and you have less and less available energy. And ultimately, this leads to disability. So this is sort of a very common model of aging, and one that we are very interested in exploring. There's a huge major public health significance. Energy is essential to health in later life. The paths are really not well characterized. We've talked a little bit about that so far today, but as you can see, there's still a lot of open questions about how energy plays a role and how that is regulated and dysregulated. And one of the keys to that is going to be, that we're interested in, is the role of mitochondria in particular. And again, the public health significance can really be shown here. You probably can't see quite the bottom of this, but this is on the bottom here is by year, and you can see both that there's not just an increase in the number of people of over 65 as you'd expect from a growing population, but they're now a larger and larger percentage of the population. So clearly, thinking about how you deal with people as they get older and how you deal with disability or prevent disability is going to be critical from a health standpoint, from an economical standpoint, all these other features. So really important. What are mitochondria? So as I said, they're the powerhouse of the cell. They're involved in producing the molecules that your body, each cell uses for energy. There are tens to thousands within each cell. You can imagine that a muscle cell may have a lot more than a blood cell, for example, because they need more energy, or brain cells, hopefully, need a lot of energy. And each mitochondrion has its own DNA, and this is sort of important for a little bit about what I'm going to say, is that you have your nuclear DNA where it comes from mom and dad, and then each mitochondria is sort of a self-contained little unit within a cell, and it has its own DNA, and that's going to be important. It does have other roles, so it's involved in communication between cells, and it's involved in cell death. So mitochondria sort of not only have this energy role, but they sort of have these other various roles intertwined with their key role in energy production. So our hypothesis is that given the central role of mitochondria in energy production and maintenance, then we think that the number of mitochondria may influence aging and age-related disease. And again, this gets to the point that there are lots of mitochondria within each cell, and each tissue is going to be composed of different cell types with different numbers of mitochondria. So this is going to vary across individuals. How do we measure mitochondria? So again, mitochondria have its own DNA. You have a nuclear DNA, so mom and dad, so you have 2 copies of your nuclear DNA, lots of copies of mitochondria. And what we can do is just take that ratio. So we can measure out the mitochondrial DNA as a surrogate for how much mitochondria you have or how many mitochondria you have. And our controller, or normalization, is always to the fact that you always have 2 of your nuclear DNA. So we can always take that ratio. So we can measure in anyone, and that's we have a built-in standard that we can use. So we've explored a number of areas in which we think mitochondria are important, and the details aren't so critical here. But here we've looked at aging. So these are 2 different graphs looking at survival over time. And what you can appreciate is that these lines, which represent the top people have the most mitochondria, and split into groups of 20%. So the top 80 to 100%, 60 to 80, and so forth. And the ones who have, let's see, is this our pointer? Yep. So here, these people have the most mitochondria. They survive the longest. These people have the least amount of mitochondria. They survive the least amount of time. And the average longevity of those 2 groups differs by almost a year and a half. So if you have really short or really few mitochondria, you're likely to live not as long. So in addition, we've also looked at, this one is sudden cardiac deaths. So there's a role apparently for Mitochondrion Atherosclerosis. So we think that if you have too few mitochondria, you're more likely to have atherosclerosis. And here we show that in sudden cardiac death, similarly, those who have more mitochondria have fewer sudden cardiac death events, and those who have less mitochondria have more events. We've also looked and shown an association with chronic kidney disease. So again, lots of aging-related diseases. And the one I really wanted to focus on a little bit was looking at cardiovascular disease. And to give you an idea, we've looked at almost 20,000 individuals. Among those individuals, we've followed them up for more than 10 years. And among those, we've had over approximately 3,700 cardiac events. They've had heart attacks or strokes. And we've looked in multiple, these are sort of some of the very big cohorts that are used across the country to look at things, risk factors for cardiovascular disease. And what we can show here, this is no increased risk. And what we can show here is that across all these different cohorts, that in fact there is an appreciable increase in risk if you have shorter mitochondria, or sorry, I keep saying shorter, thinking telomeres in my head, but no, fewer mitochondria. And actually there is a relationship between the two, but I'm not going to talk about that. There is some suggestion that the amount of mitochondria also correlates with the length of telomeres, something we're looking into. This is a complicated table. I don't want to go into the details of it. Just to point out, what we've done is taken the standard risk calculator for those who should be put on a statin, something like Lipitor, to lower your bad cholesterol. And what we can do is we can say, we'll take the current risk calculator and just add in the amount of mitochondria that you have and say, does this help us decide who should or who should not be taking a statin drug? And on top here, these are people who don't have heart attacks, and these are people who do in our study. And so these are people who would be given a drug. If they don't have a heart attack, we want fewer people in the red box. These are people who would not take the drug, so we want more people in the non-taking drug if you're not going to have a heart attack. And we want people, oops, that was interesting. How did we get out of that? It's back in there. Sorry about that. Apparently if you hold it down too long, you run into problems. And right here, these are people who have heart attacks. We want more of these people to take a drug, and we want fewer people not in this group not taking a drug. We see, in fact, that when we take the standard risk calculator and just add in the number of mitochondria, that we can actually improve both the sensitivity and specificity. That means that more people who need the drug will take the drug, and fewer people who don't need the drug will not take the drug. So in conclusion, mitochondria is mitochondrial copy number. So the amount of mitochondria is a novel independent risk factor for lots of aging-related diseases, in particular overall mortality, chronic kidney disease, and in particular, I think, cardiovascular disease. And what we've done, which I'm not going to present the data, we've done something called a Mendelian Randomization. And this is really asking the question of causation versus correlation. So you can imagine there may be a very important biomarker. So your good cholesterol is an example of this, where we measure it, it's important. But in fact, if I change the amount of good cholesterol, it does not change your risk for disease. Bad cholesterol, on the other hand, if I change the amount of bad cholesterol, if I lower that, I decrease your risk for disease. And so we want to ask that question here. If I increase the amount of mitochondria you have, does that actually lead to a better outcome? Or is it just something that if I measure it, it's going to be predictive, but if I change the levels, it's not? And again, the preliminary data suggests, in fact, that it is causal. So that if I increase the amount of mitochondria that you have, that you're likely to have better outcomes. And so this then, of course, obviously leads into the thought of, are there drugs out there? And there actually are some FDA-approved drugs that already increase mitochondrial copy numbers. So exciting areas for future research. And I'll take questions. Oh, probably put my acknowledgements there.
Hi, Chip Rose from the NIH. I was wondering, what are some natural ways to increase your mitochondria?
So we are looking right now at exercise as one of those. So obviously exercise is always good for you. But there's a trial we have where we had individuals who are measured at baseline, and then they have a 6-month exercise intervention. There does seem to be some suggestion that, in fact, exercise does increase the amount of mitochondria. And again, we're looking in blood, so we're not looking in muscle. It'd be nice to get some muscle biopsies, but not everyone's always willing to give them. So even in blood, we do see at least preliminary data suggesting that there is an increase with exercise. So that is one way that you could potentially do it.
Kevin Soden with ShareCare. I wonder, is there any practical application of this? Can I measure this now in my clinical practice, for instance? Or, and then if I do that, will it help people maybe change their behavior, saying, gee, you're in a really low count?
Right, two tough questions. So the first one is, in terms of a clinical test right now doesn't exist. We'd love to develop one. I think the problem here is developing standards. So far, all our measures are relative to everyone else in our group, but it can definitely be done. The methods that we use are actually very simple. It's a quantitative PCR. It's a test that costs a couple dollars. So theoretically, if a company goes out and develops the assay and the standards, it's something that could be relatively easily implemented in the clinic. And I think we have some pretty good data, at least for cardiovascular disease, that it actually does help your risk calculation. Whether it changes behavior is always another question. I think you could tell, I mean, people have a heart attack don't always change their behavior. I think it is useful, again, from I think our statin example, where I think it would be used in clinical practice, where you could say these people really shouldn't be on a statin, and these people should. And there's sort of an argument that maybe everyone should be on a statin, but there are side effects that people sort of tend to ignore. So I think it's important in both directions.
McIntyre, NIH. I don't know if this has anything to do with mitochondria, but does nicotinamide mononucleotide affect mitochondria? And what is it so great for? I mean, it's kind of got a lot of hype.
I do not know whether it affects mitochondria. I haven't actually, in terms of,
in
measured levels, I'm not sure the correlation. All right, thank you.